整備者でございます。 CEO のミスターデスピール氏をお迎えしておりまして、マクロ解剖や分子分析で活用可能な画期的腫瘍自動検出法でございます。
Good morning, ladies and gentlemen. I'm Des Speeder, CEO of PathXL. We are a software company. We specialize in digital pathology. We're from Belfast in Northern Ireland. Our company was founded in 2005. You may be familiar with digital pathology, but just to mention, digital pathology helps to replace and support in different ways the microscope as a device for viewing pathology images. And what digital pathology enables is modern workflows. We put around that for collaboration. And what I'm going to talk about today is a new phase, which is about analysing the images once they're captured in a way that's difficult to do manually. The company originally spun out from Queen's University in Belfast. Professor Peter Hamilton founded the business, a pioneer of digital pathology back to about 1992 when he lived and worked in Tucson, Arizona, the University of Arizona. We also have a strong validation partnership with the Belfast-based Centre for Cancer Research and Cell Biology, and our new software, which I will explain, has also been validated by AstraZeneca in England, which gives us an independent and a pharmaceutical company view. This slide gives you a flavour of some of our existing clients, just as a point of reference. A series of pharmaceutical, CRO, diagnostic, university, hospital groups in Europe and USA. And my reason for being here this week is we believe that the nature of our technology has benefit, which may be of use here, particularly given the relative shortage of pathologists. There's a shortage of pathologists worldwide, and the ratios in Japan are somewhat more acute than a number of other countries. In terms of digital pathology and the software we provide, this set of circles shows in green aspects of education and training, in the maroon colour aspects in research, and also coming up in a moment or two in blue aspects in support of clinical diagnosis. I'm here today really to talk about the image analysis in research, which is our exciting new application with major potential. And one of the key areas that we are focusing this technology on first is the Microdissection Process as part of the whole personalised medicine DNA profiling. The set of pictures at the top represent the manual process with manual markup, and there's a short video coming up which will show this better than I can explain. And the set of slides at the bottom shows the revised process using digital pathology with the automated computer markup. And the question is, why this? Well, certainly the shortage of pathologists poses pressure in terms of timescales, turnaround, pressure of work and all that goes with the variability of pressure of work, and also the whole aspect of consistency and quality. And as we talk to clients, we're currently engaged with something like 95 different clients from major pharma, CRO, diagnostic companies in USA and Europe, and something that's becoming clearer and clearer to us is the need for more consistency. These are two academic publications within the last 6 months, nothing to do with PathXL, that highlight the risks of variability in manual pathology, and in particular overestimation of tumour percentage and the potential impact that can have on clinical outcomes. So our software, the green line is an indication of the software marking up the area of tumour in this particular tissue type, and also identifying the percentage of tumour within the boundary and also the percentage across the whole slide. This slide shows a comparison, if you like, between a simple manual pathologist markup and on the right the software markup, which, as you can see, can be more precise, and also within the tumour boundary show this heat map where the areas in red are the areas of high concentration of tumour. What our software can do as well is allow the pathologist with QA to move in and out to highlight areas of particularly high tumour concentration to focus on those, especially for microdissection. So in summary, as a company, digital pathology has moved beyond being pioneering to getting closer to mainstream adoption in general. We believe we have an exciting new application here which will be part of helping to take digital pathology from just collaborating to analysing and helping to automate and improve consistency, and in so doing improve the overall clinical outcomes. We believe this type of software is bound to become ubiquitous across the world. What I'm going to do in a moment is just to show a video, if you will bear with me.
PathXL is focused on delivering top quality products that make a real difference in the pathology lab. TissueMark is a new range of developments from PathXL. It's designed for the automated detection and annotation of tumour tissue on digital slides. It draws the tumour boundaries automatically and then gives you a solid and consistent number for the percentage of tumour cells in the sample.
The molecular evaluation of tissue samples is absolutely central to discovery in the pharmaceutical industry for discovering new drugs, in the diagnostics industry for identifying new biomarkers of disease. TissueMark is a really important development that allows us to fast-track tumour annotation, identifying key areas of tissue samples from which DNA and RNA can be extracted. And this really speeds up the process of identifying new markers of disease and developing new drugs. Pathologists traditionally mark up slides in a very awkward way using a marker on a microscope. TissueMark allows them to review slides much more rapidly, much more efficiently and cost effectively than they currently can achieve. The PathXL TissueMark product is very easy to incorporate within laboratories. Very intuitive, very easy to use, and allows laboratories to increase the numbers of tissue samples that can be reviewed for large and for small research projects. It also enables them to access pathologists and pathology skills from anywhere in the world. This will allow industries and academic organisations to accelerate national and international research programmes, significantly improving our molecular understanding of disease and enhancing the development of personalised medicine.
So tissue annotations and subsequent macrodissection of those tissues for molecular analysis is a very important component of modern molecular diagnostic laboratories, and is also a big help in translational research, because there are more and more cases coming to our laboratories. This is putting a lot of strains in tissue pathologists that need to do this task. For that reason, I think it's very good news that we have systems that are helping the pathologists to do this in an accurate and in a timely manner. So I would say that for these systems to be useful, they have to be automated. They have to have a very easy software that allows minimal handling, and primarily a system that allows me to do these annotations from somewhere else, even if I'm not physically in the laboratory. With all these characteristics, I'm sure that this system is going to make a change, both in the way we practice molecular diagnostics and also in the way we do research in molecular pathology laboratories.
TissueMark is fast, accurate and highly consistent. You also get all the benefits of digital technology and workflow management.
So in conclusion, just to say today we have, we launched this new software in November at the Association of Molecular Pathology Conference in Phoenix, Arizona. And today we have, in terms of tissue types available, lung, colorectal and breast cancer. We're currently actively working on prostate, ovarian melanoma, which will be launched quite soon. very conscious from this week of the prevalence of gastric cancer here, and we're in discussion with partners here who can help us take that and accelerate that into our portfolio as well. This presentation is focused on the macrodissection use of this image analysis. There's also a biobanking use for screening in biobanks, and one of the top 3 cancer centres in the USA is currently contracting with us, and we'll be the first client, and that's the use that they will be putting it to. And we're also clearly very conscious of the potential in clinical, in the context particularly of prostate screening, in terms of the amount of work and pathology effort that is involved in that we can help support. Thank you very much for your attention. Arigato.